Hello, everyone, and welcome back. This time, we'll talk about the process of acute inflammation. If you accidentally cut your finger, besides bleeding, the wound would become red, warm, swollen, painful, and may form pus if the wound is dirty or become infected. You may ask how these local manifestations develop. Learning about the process of acute inflammation will tell you the answer. The process of acute inflammation has three main steps. First step is recognition of microbes, necrotic cells, and foreign substances. This step will activate host responses to injury, including vascular reactions and cellular events. There are the two major components of acute inflammation. Vascular reactions involve changing of the local hemodynamics and increased vascular permeability. The main purpose of vascular reaction is to deliver leukocytes and plasma proteins to the extravascular space. Cellular events mean that leukocytes transmigrate through the blood vessel wall, accumulate in the inflamed area, and exert their functions. We'll talk about the process of acute inflammation through 3 sessions. Today, we'll focus on recognition and vascular reactions. After this session, you are expected to know what does the process of acute inflammation involve? How do we describe the hemodynamic changes? What are the mechanisms underlying increased vascular permeability? First, I'll introduce recognition. When tissue is injured or infected with microbes, for example, caused by a cut on the skin, the products of microbes or dead cells would be recognized by the tissue sentinel cells, mainly macrophages, but also dendritic cells, mast cells, and other cell types through cytoplasmic and plasma membrane receptors. These cells produce cytokines and other inflammatory mediators to trigger vascular and cellular reactions. Inflammatory mediators are chemical substances that initiate and regulate inflammatory reactions. Besides produced by host cells, inflammatory mediators can come from plasma proteins. For example, toy-like receptors are important microbial sensors. They can recognize products of bacteria, viruses, and other pathogens. Toy-like receptors are located on plasma membranes and endosomes and are able to detect extracellular and ingested pathogens. Recognition of pathogens by toy-like receptors stimulates the production of cytokines. Next, let's look at vascular reactions. Local hemodynamic change consists of 3 steps. The first is the change in local perfusion with transient vasoconstriction of arterioles. Here is a picture showing microcirculation, arterial capillary bed, and postcapillary venule. Arterial constriction leads to decreased blood flow in this microcirculation, resulting in brief pallor of the inflamed area. This process only lasts for several seconds. Then arteriolar vasodilation occurs, followed by engorgement of the downstream capillary beds, leading to an upsurge of blood flow. This process is similar to arterial hyperemia. Therefore, the inflamed area is red and warm. The third step of hemodynamic change is increased microvascular permeability, slowed blood flow, eventually blood stasis. This process causes vascular congestion. The enhanced permeability of the microvasculature leads to extravasation of protein-rich fluid through vessel wall to form exudate. This extravasation lowers the intravascular colloid osmotic pressure and accelerates water outflow from the blood. This makes the blood more concentrated, thereby increasing blood viscosity. Blood flow is slowed down and may even stop. As the blood flow becomes more sluggish, leukocytes migrate from the center towards the periphery of the blood vessel and accumulate along the vascular endothelial surface. This process is called leukocyte modulation. Modulated leukocytes adhere to the endothelium, facilitating their transmigration into the interstitial space. One of the mechanisms of vasoconstriction and vasodilation is neural reflex, but it only exerts a transient effect. The main mechanism is the function of inflammatory mediators, which last for a relatively long time. As mentioned earlier, vasodilation is quickly followed by increased vascular permeability and subsequent extravasation of protein-rich fluid. Here, I list 4 mechanisms responsible for this phenomenon. All of them may take place simultaneously or successively. We'll go through them one by one. Endothelial cell contraction resulting in intercellular gaps in post-capillary venules is the most common mechanism. It occurs rapidly after exposure to histamine, bradykinin, leukotrienes, and many other inflammatory mediators. This process only lasts for a short time. Hence, it is referred to the immediate transient response. Another mechanism is increased transcytosis through the endothelial cells. Transcytosis means protein-rich fluid moves across endothelial cell via intracellular channels formed by fusion of vesicles. After exposure to certain mediators, such as vascular endothelial growth factor, the number of vesicular channels and the caliber of vesicles both increase, leading to enhanced transcytosis. Endothelial injury causes endothelial cell necrosis and detachment, further exacerbating vascular permeability and subsequently vascular leakage. Direct damage to the endothelium is encountered in severe injuries, for example, in burns and some infections. Leukocytes that adhere to the endothelium during inflammation may also injure the endothelial cells by releasing toxic mediators and proteolytic enzymes. In most cases, leakage begins immediately after the injury and persists for several hours or days until the damaged vessels are thrombosed or repaired. Lastly, we have leakage from the new capillaries formed during the inflammatory reaction. The endothelial cells of the new capillaries are immature, therefore their intercellular junctions are loose, resulting in increased vascular permeability. This remains until proliferating endothelial cells mature sufficiently to form tight intercellular junctions. Now, it's time for a summary. We used a cut on the skin as an example to illustrate the process of acute inflammation. The cut causes tissue injury and infection, which can be recognized by the tissue sentinel cells such as macrophages. These cells produce cytokines and other inflammatory mediators to trigger vascular and cellular reactions. Vasodilation and increased blood flow cause the wound to be red and warm. Vasodilation is quickly followed by increased microvascular permeability and subsequently slowed blood flow, leading to inflammatory exudate, which causes the swelling of the wound. We talked about 4 mechanisms underlying increased vascular permeability. Among them, endothelial cell contraction is the most common mechanism. You would ask, what is the principal component of yellow pus in the wound? Learning about cellular events will tell you the answer. Next time, we'll talk about cellular events. Here are the references. Thanks for joining us.